Hi, and welcome to all who joined on today's webinar. I hope that everyone is doing well. I'm Eva Żarnowska, an application scientist at Bruker Fluorescence Microscopy Business Unit in Madison, Wisconsin. It is my great pleasure to introduce today's speaker, Dr. Roberto Araya, an associate professor of neuroscience. He has a joint appointment in the Department of Neuroscience and at the University of Montreal and the CHU Saint-Justine Research Center. This joint appointment was established to develop translational work in autism spectrum disorders within the Child and Brain Development Research Partnership between these two Canadian institutions. Dr. Araya completed his graduate studies at the Catholic University in Chile and University of Bonn in Germany. For his postdoctoral work, Dr. Araya joined the laboratory of Dr. Rafael Yuste at Columbia University in New York. Dr. Araya is passionate about dendritic computations, specifically how dendritic spines control the processing, storage, and integration of synaptic inputs. He's extending these questions to neurological disorders to uncover abnormal neuronal and circuit elements via a complex approach that includes state-of-the-art, custom-built in vivo and in vitro two-photon microscope setups with holographic simulation capability, as well as electrophysiological, structural, genetic, and molecular tools. Before I hand the mic over to Dr. Araya, I have a few housekeeping items to cover. We would love to hear from you during presentation. If you have a question for our speaker, please feel free to send it through Question tab. We will be answering questions at the end of the session. However, if you have a burning question, please feel free to raise your hand also via Question tab, and your question will be answered immediately. We would appreciate your sharing today's webinar on your social networks. Please do not forget to add hashtag BrukerNano to your post. So let's start then. The title of today's presentation is a spike timing dependent plasticity rule for dendritic spines. Roberto, over to you.
Hi, thank you very much, Eva, for the invitation and for organizing this. It's really a pleasure to be here today and to tell you guys a little bit of what we've done in the last years, trying to understand the spike time independent plasticity rule for dendritic spines. Do you hear me well? Is it fine? Anyway, so so this work was done, as Eva mentioned, in University of Montreal and at the CHU Saint-Justine Research Center here in Montreal. And so let me just start by briefly telling you, Eva mentioned that what are my interests, but I work primarily in a structure called the cerebral neocortex. And the cerebral neocortex is the thin layer that covers the brain that it is involved in higher functions such as sensory perception, motor commands, spatial reasoning, conscious thought, and human's language. And it's a very, as you could tell, important structure. And the picture I'm showing you here is a picture I took from my kitchen window after a snowstorm in Montreal. And that was one of the first years when I arrived in Montreal from New York, which I thought it was already cool. A cold city, but Montreal is really beautiful, but very cold. And this snow was packed in my window, and it looked really, really like the cortex in my mind. And my son asked me, he was a very young kid then, asked me what was my work, what is that I work on. And so I grab a pen and I just draw all these neurons. Can you see my pointer? I guess I cannot ask questions because nobody will answer me, but okay, great. So basically I grab a pen and I drew these cells that are the cells that I work with, which are called the pyramidal cells. I mean, my son was excited about me painting the house and drawing in windows and walls, not really on the science, but I thought it was pretty cool to show him. And this is a sort of a representation of the cortex. The cortex has 6 layers. The first layer is the layer that basically receive feedback connections. Is primarily axons, contacting neurons from different cortical areas. And then you have the somas, which have this pyramidal shape. That's why they're called pyramidal neurons. And those somas are located in different layers with between layer 2, 3, 4, 5, and 6. And depending where this soma is located is how we call that neuron, either layer 5, layer 6, layer 2, 3, layer 4. Okay, but you can see that the layer 5, for example, in this case, the dendrite pretty much covered the whole cortex. So they're receiving information not only from layer 1, but from all other sections. And these neurons in general are very important because they receive feedback information or contextual information and sensory information in their proximal dendrites. As you can also see probably from the drawing, I went into the trouble of making these tiny little protrusions here, which are called dendritic spines. And these are basically maybe here. Okay, let me just also say that these pyramidal neurons are the most abundant neurons in the cortex. They represent more than 70% of the neurons. And they are very important. In particular, layer 5 pyramidal cells are the main output neurons of the cortex. So they receive sensory contextual information, they make associations, and they decide, depending on the amount of voltage that is generated at the soma, whether they send the information out or not. And this is now a real image of a pyramidal cell. And with a mouse model, with a mouse we have in the lab that express EGFP in pyramidal cells, in particular in layer 5 pyramidal cells. And you can see the pyramidal shaped soma that is similar to the drawing here. And then you have these basal dendrites, which are, or proximal dendrites. And then you have the thick apical dendrites that go all the way up to layer 1, forming a tuft that receives information from other cortical areas. But in this image, I hope it's clear and you can see it clearly that there is these tiny little things that are popping out that are these little lollipops or beautiful looking structures that are dendritic spines. And dendritic spines were observed by many researchers back in the 1800s, but using the Golgi method. But everybody thought that those structures, which are here represented in this drawing, were an artifact. So as a consequence of that, they were making or drawing the dendrites as completely smooth without any protrusion. But the first that proposed and said that these structures are not just artifact of the staining technique, but they are real structures that connect neurons, that serve to connect neurons, was Santiago Ramón y Cajal, that proposed that in 1888. As you know, dendritic spines, dendritic spines are very, very peculiar structure. They have this very thin neck connected to the dendrite, this small head connected to the dendrite by this slender neck. And they have different shapes and morphologies. And this is, with the advent of nonlinear microscopy, we have been able to image and manipulate live dendritic spines, which is the core of what I will present you today. But in addition, techniques like electron microscopy, where you can have the ultrastructural resolution of dendrites and spines and pretty much every single structure you want, you can reconstruct the spines and look at the postsynaptic density, which is this molecular structure that is sort of the fingerprint of an excitatory connection. So here, the reconstruction of the postsynaptic density, the place where excitatory connections occur, is reconstructed, and you can clearly see that excitatory connections only occur in dendritic spines heads, and the shaft of this piece of dendrite, right? So basically the dendrite, it does, in pyramidal cell, don't receive excitatory connection. So this tells you immediately that spines are the main gateways for the entrance of excitatory inputs in pyramidal cell. So in my, if you will, micro approach is, if I really want to understand how the brain works, at least in my interest is to go to the minimal unit or the gateway where this information is being received, and let's try to understand how those devices transform synaptic information to try to understand how the cell can process that information and ultimately send a signal to other neurons in the network. Okay, so In my lab, we have been actively working, manipulating or working with dendritic spines to study these three key processes, which is synaptic transmission, the integration, or how inputs are being, integrated. If you receive multiple inputs in the dendrite, how they add up together, and then the storage or synaptic plasticity, which is what I will talk to you about in this presentation. So, as you know, synaptic plasticity it's a very well-known process, and it's ironclad that dendritic spines can undergo structural remodeling and are the preferential site for the induction of LTP, or long-term potentiation. I will call it LTP, and long-term depression. And also, you probably already have very well imprinted in your brains that these morphological changes include spine head enlargement when you trigger a process like LTP, or in the case of LTD, spines can shrink their shape or their size, and sometimes they can disappear. And these two processes, the increase of the spine head and increase the amount of AMPA receptor and the amount of glutamate receptors, increasing the synaptic weight, and the same for LTD, but in the contrary. So basically, you have a reduction of receptors, the cell, the spine shrink. Those processes have been observed in vivo and also in vitro. In vitro, you can use two-photon uncaging, and you can use certain conditions here, which I highlight here, which we can discuss it later. But you can trigger in a matter of minutes by just doing high frequency stimulation and playing a little bit with the divalent concentration of magnesium to try to maximize the activity of NMDA receptors to trigger LTP or to trigger LTD. So we can manipulate those single spines by the use of a two-photon microscope and trigger this process of depression and potentiation. And as I said, in vivo, those changes have been observed in hours, in a matter of hours, 1 or 2 hours or 3 or more hours during learning paradigms. You can see that there are some morphological changes of the spine, right? And the other important thing for me to mention is that there is very good correlation between the size of the spine head and the postsynaptic density and the amount of AMPA receptors. So this is sort of what we tend to know in very general terms about the structural and functional arrangement of spines and how that translates into changes in synaptic weight. So, and this is also something that I will say is not too controversial, but that the plasticity or the induction of LTP and LTD are thought to be the underlying mechanisms of learning and memory. So in other words, if we really want to minimize or go to the minimal functional unit of memory, for sure for plasticities and dendritic spine, but it's likely that for learning and memory, that could also be the case. And there's a very good experiments that have shown some very strong correlations between the increase in or the morphological changes of dendritic spines and the buildup of memories in the brain. So that's sort of the, if you will, the minimal expression of plasticity and possibly learning and memory is a single dendritic spine. So this little pocket, these little lollipops and devices are incredibly relevant for who we are and how we build identity by building engrams in our brain. And not surprisingly, in neurodevelopmental disorders, the substrate that is mostly affected are in let's say, for example, in autism or in Alzheimer, are these little dendritic spines. And they act as electrical devices. As you know, the dendrites are cables, they have high impedance, they can just control the engagement of voltage-gated conductances, the activation of NMDA receptors. So it's a very interesting electrical pocket, but also a biochemical pocket. So in other words, you can trigger a series of molecular cascade that ultimately will lead for the strengthening of that synapse and forming the process of plasticity. Okay, so I think I always like to tell the real history behind stuff. So why I really landed on LTP and studying plasticity, LTP and LTD, is because when I was doing my postdoc, I found this very surprising results, I would say, that if I was doing uncaging in different spines, even if they were very near to each other, and with similar head sizes, so assuming that they might have similar amount of glutamate receptors, the strength of the synapses were completely different. So in this case, for example, this spine, as you can see, has a longer neck. The response that I record at the soma, let's say 30, 40 microns away from activating that spine, is 0, but that nearby spine generates some depolarization. So we did that with uncaging, but also with minimal synaptic stimulation in a recent paper in 2014. And we saw pretty much the same, that the causal signals here are the spine is activated electrically, and then it can trigger a clear causal signal, but no voltage responses. So this was very puzzling for me, because that tells you that there's a big repertoire of spines that aren't contribute almost nothing to the somatic voltage. So that's the genesis of why I went in trying to study plasticity in the system, because I wanted to know if this repertoire of spines that are less relevant for the network might get more relevant by just having some sort of plasticity process, which in this case I thought it could be a twitch of the neck or something that might change the impedance. And at the beginning I was thinking primarily on purely electrical terms, but you will see in this presentation that it's not that, it's actually that and something else. Anyway, so looking at different protocols, high frequency stimulation of spines, and playing also with the divalent cation ions. So then I realized that there was nothing known about the rules of spike time independent plasticity at the level of single spines. And I'll tell you immediately what is spike time independent plasticity. The majority of you already probably know about it, but I think we cannot talk about spike time independent plasticity without mentioning this Canadian neurophysiologist, Donald Hebb, that in his book, The Organization of Behavior, that he published in 1949, he provided the formal postulate of the neural mechanisms of learning and memory. And in his postulate, he said that the coordinated activity of a presynaptic terminal and a postsynaptic neuron would strengthen the connection between them, and conversely, an uncoordinated activity between the synaptic partners would weaken their synaptic connection. So neurons that fire together wire together. And then, he had this model that basically when an axon of cell A is near enough to excite a cell B and repetitively or persistently takes part in firing it, some growth process or metabolic changes will take place in one or both cells, such as A's efficiency as one of the cells firing B is increased. So in other words, he's just talking about some molecular changes and the importance of this coordinated or uncoordinated activity to trigger an engram or storing some information. Well, STDP is precisely that. So spike time independent plasticity is a process in which the order and the precise temporal interval between presynaptic and postsynaptic spikes determine the sign and magnitude of the long-term potentiation, the LTP or the LTD. And this is a classical plot that you might have seen many times before, where this is a connected pair. And it's important because, when the presynaptic cell spikes within 20 milliseconds before the spiking of the postsynaptic cell, you have a potentiation. But if you invert that and then you have the postsynaptic cell firing before the presynaptic cells, you have depression. So in this, this will be the coordinated and the uncoordinated. So you have, in this case, a classical bidirectional Hebbian STDP rule. And this is CA1 cells, but also in cortical pyramidal cells. The same here, you see how the strength of the connection between two cells, if the pre precedes the post, then you have potentiation. If the post is before the pre, you have depression. And nothing happened if it's just a pre only or the post only. And so this was really has been influential since these experiments were out. In 20 or more years, there has been influence biology and neuroscience experimentally, and also modeling neuroscience. So it's a very important role. But and this is going back to the cells that I really care about, layer 5 pyramidal cells, and this is this beautiful work from Jesper Jostrom from McGill University and Mike Heuser. This is when he was at UCL. And this is a group of, he patched multiple layer 5 pyramidal cells and to find connected pairs. And when the presynaptic cell fires before the postsynaptic cells, and he repeat that, you can see a potentiation of the response. So the pre before the post triggers this strong potentiation. So what I really wanted to go into is to try to understand what is the structural organization of the excitatory input that is capable to generate this process. As you know, the data I showed you before, until we jump into really trying to crack this STDP rule at the spines level, it was primarily connected pairs experiments and electrical stimulation of axons to try to activate synaptic contacts or dendritic spines. But there was some attempts, but not really a thorough characterization of the whole process of the learning rule of the STDP. And so the idea is what happens. So if you have these connected pairs, where are those inputs being impinged, and how they are organized, and how this organization is responsible or not for triggering either time-dependent LTP or time-dependent LTD. Okay, so the good thing is that with uncaging, let me see. Yeah. The good thing is that we can do this because we can do this at will. Because with uncaging, we can mimic the presynaptic release in any single spine that we want. And so if you patch the cell, the postsynaptic cells, and you trigger backrotating action potential, then you can just time, you can trigger an STDP protocol either to trigger LTP or LTD at will. And the question will be then, if we can do that with uncaging, is what happens if we activate a single spine, mimicking the presynaptic and activate the postsynaptic? Is that rule for single spine following a bidirectional Hebbian STDP rule or not? What if we activate two spines together? And what if we start to separate them out? Is there any change in the learning rule by just playing around with what spines we are specifically activating? So, and with that, we can also not only learn about the patterns of activity of the structural organization that supports I will call T time-dependent LTP and T LTD as time-dependent LTD. And we can look at the morphological changes of the spine, and also we can look at some of the molecular changes and molecular changes that occur in the spine that might lead or not to the induction of any of these changes if we see them. So as I said, I'm going to go brief here, but we use two-photon uncaging of glutamate. It's a cage glutamate. It's a commercial one that we use is MNI glutamate. And then what we use is we use a fixed concentration with bath apply, a fixed concentration of cage glutamate, and then we deliver short pulses of light, 2 millisecond short pulses in each spine to trigger a synaptic response or to mimic the presynaptic release. And as I said, here is the cage glutamate of these squares, and then only on the focal spot we will be able to generate and move and release the cage from the glutamate and activate a single spine. And we have done a lot of work in trying to titrate down this and have a process that is very reliable at triggering synaptic responses in a single spine without spillover to nearby spines. All right, so with that, we can just trigger the EPSP or the mimic the presynaptic release before we generate an action potential with a patch in that cell or after. So we can really play the timing here at will. All the experiments I'm going to show you are done in basal dendrites, which, as I mentioned maybe briefly, but these are very important dendrites because they receive the sensory information in the cortex. And the apical tuft here receives a contextual or predictive information. So this is sort of the window for sensory information in the cortex. And so the setup, and I hope if you want to get more details about our setup, here we have the reference for a recent paper that we published where we describe this cost-effective microscope that we can do nearly simultaneous two-photon uncaging and imaging of activated spines. So the microscope is not very complicated. It's a two-photon microscope with a femtocircular pulse T-sapphire laser, focal cells, a series of lenses to change the beam size to accommodate for the different objectives. And then we have a Bruker scan head and Bruker software that with that we control uncaging and imaging. But we have only one pair of galvo mirrors, and so we really wanted to be able to do almost simultaneous uncaging and imaging. So in this particular case, we didn't have much money to have another set of galvos, so we couldn't afford that. So what we came out with, and with the great help of Mohamed Nazir, so we actually added a Bruker switch box, which is a high-speed electronic module that allows us to switch between imaging and photo activating or uncaging mode in less than 2 milliseconds, right? This module is fully integrated with the PrairieView software, and it consists in a series of multiplexes that integrate and route the imaging and uncaging commands to the galvos or the focal cell controllers. And the advantage of this is that we use only one laser source and one set of galvos, and we can actually image the spine, uncage, and go back and image the spine, and go around that with a system that it's not cheap, but it's not incredibly expensive. And I have to say, this is one of the great reasons why I will always recommend people to go to Bruker for their software and their really modularity of this. I know other companies are doing similar stuff, but in my particular case, it really helped me to get the work done. Anyway, so here, the classical line scan mode that you can do in the spine, where you can uncage and do a line scan of the spine, before and after. But this is sort of, I guess, more relevant for this talk today, which we use the switch box to go from an imaging mode. So this is sort of the in time. So here's I'm taking an image of the spine, region of interest. So we wanted to have information about the data of the spine. We're paying the price a little bit about the temporal aspect, because we don't have like resonant galvo mirrors or just regular galvos, but we have here, we're scanning the spine. Then we rapidly use the switch box to go into the uncaging mode. We increase the laser because power. And by the way, I have to say that we use the same wavelength for uncaging. We use a very low power at 725 for imaging that doesn't trigger any uncaging. And then we rapidly, with the focal cell, up the power of the laser, we can generate uncaging. So we don't have to switch, or then we can just go around with just using one laser. So we can image, then we trigger the uncaging spot, and then we go back to imaging. So we perform 4 or 5 images, or 4 images, 1 before and 5 after the uncaging. Here I'm showing different images, the uncaging response. So now we can get this nearly simultaneous, but not exactly simultaneous, but nearly simultaneous imaging of the activated spines while we're recording the synaptic responses at the soma. All right, so sorry for showing a picture of me, but this is the latest picture I have from the setup. It's, as you can see, it's not awfully complicated, and I still have the old sticker of Prairie Technologies, but I think Bruker will be very happy, because they have a full page on a Chilean newspaper. This was the picture that the Chilean newspaper used to flag our research. And here, if you look at the print version of the paper, it's absolutely clear. It says like Prairie Technology. So maybe you can put that on, or I'll send you the link for that, guys. But anyway, to say that this fantastic microscope is not too expensive, and it allowed me to do marvelous things. Okay, so let's go back to the science. So so how we can trigger STDP. So here we use a pairing protocol. The pre precedes the post, and the pre in this case is the two-photon uncaging. So we are patching a cell, loading it, looking for the spines, and then we select, let's say, that particular spine, and then we uncage, just uncaging at a low frequency to get the fingerprint or the synaptic strength of that connection, of that spine, sorry. So we get what is the EPSP and the morphology of the spine. Then we trigger a pairing protocol, which is in this case 40 repetitions, and we try different timings between the pre, which is the two-photon uncaging, and the back-propagating action potential or the post activity. So we did like 7 milliseconds and 13 milliseconds, and we repeat this 40 times, and then we go back as fast as we can to look at the spine, and I just engage at low frequency to see the voltage contribution of that particular spine. And we ask the question whether there was a potentiation, depression, or whatever, and what happened with the morphology of the spine. And as I said, I'm just like looking at 2 particular time points in this process. All right, so this is how one experiment looks like. So this is a particular spine that we trigger TLTP at this particular timing of plus 13 milliseconds. And this is the average of 10 uncaging responses. You see that they are reliable. There's some variability, but they're pretty reliable. These are very small responses, less than a millivolt. And then what you can see is that 3 minutes after this process, or in this case, we image 3 minutes after, and we look at the electrophysiology, and you see a significant increase in the strength of that connection, of that spine, and then, and then a significant change, or like a change in the neck length. So the neck shrunk, and then the spine basically retracted, and the spine head did not change significantly. But you can see that the APSP pretty much remains up for 40 or about 40 minutes that we were able to image without compromising the health of the dendrite or the spine. So this is sort of how the temporal course of the experiment goes. We're imaging the spine. We're going to show you later that we also image the calcium, and then of the dendrite on the spine, and then we look at the electrophysiology in the soma. This is the experiment I just showed you. And when we plot all the experiments together over time, what you can see at this particular time, pre-post timing, we see that the EPSP goes up after several minutes. It remains up. The neck shrunk. There's a shrinkage of the neck, significant shrinkage, but there's no changes in the spine head. And that might be controversial to what I mentioned to you before, but I think what we uncover here is a novel earlier stage in the process of memory, or memory plasticity, that is prior to the consolidation, if you will, changes that happen at the spine that basically grow and basically remain as a stable unit of plasticity. All right, so this is the message. I think, you have a significant shrinkage of the spine neck, no changes in the head, and there's a potentiation at that particular timing. When we look at a different timing, let's say plus 7 milliseconds here, we saw that this kind of want to go to a plasticity, but not really. So this is like not significant changes in any of the parameters that we observe. But only at this particular timing, there's a very successful time where we can trigger, when we activate a single spine, plasticity and morphological changes. I forgot to mention that this plot here is when we took the max change in plasticity, in uncaging potentials, and the concomitant changes in spine morphology at the same timing. So this is also another way to show you the spot where we have the max effect, either up and down, and it pretty much follows what I said. There's a potentiation, neck shrinkage, nothing happened with the head, and at this other timing, which is pre-post, there's no induction of plasticity. So that was very interesting. So as you can see, we're already uncovering the same sort of plot that you saw with connected pairs, but now at a single spine level. And these experiments are incredibly tough. So this whole project took about 7 years, and people that work in spine uncaging know exactly what I'm talking about. So it would be ideal to test many other time points, but it's a really, really, really tough experiment. Anyway, so we have here a situation where in the same STDP plot where we have this bidirectional STDP rule, right? We see that there's this timing that trigger LTP very effectively. We change morphological changes. This other one, nothing. So what happened now if we do the inverse, if we activate the post before the pre? All right, so the protocol, similar, but now we generate the backpropagating action potential in the postsynaptic cell before the uncaging. And we follow this protocol, we look at the spine, morphology, we look at the EPSP, and we tested 2 different time points. what we observe is that if we activate or uncaging that spine before in the induction of the STDP protocol, in this case, the TLTD protocol, the depression protocol, we see that the EPSP or the contribution of that particular spine is about, it's about less than a millivolt, but like 0.5 millivolts. Then we trigger this pairing protocol that trigger depression, and what we observe is that there's a significant reduction of the EPSP, but there's no morphological changes of the spine. Again, this is not saying that the morphological changes will eventually occur if you have a situation where you keep probably trying to depress that spine. So this is, as I said, maybe a prior stage before the full shrinkage and disappearance of the spine. But anyway, so we're very happy because we were able to trigger depression. And when we look at the transition or the timing of how this process goes, this is a depression. So it basically starts to appear after several minutes of the induction protocol. There's no changes in the morphology of the spine, which are also depicted here.
Roberto?
So the message, yes, sorry.
I have a question. I think it's about LTP still. At what time point was the neck length measured after the pairing?
Right. So the neck length. So there's a limitation on how fast you can image after the pairing protocol, right? So what we want to go is to do some C-stack imaging of the spine to be able to really assess as best as we can the morphology of the spine. So I think the fastest we were able to do it is about 2 minutes. Because you have to take into consideration we finish the protocol, and then the whole protocol, counting the time of the protocol, and then after you finish the protocol you can go immediately and image. But maybe the plasticity occur while you were doing this pairing, so it's very hard to know exactly when you trigger the morphological changes. And maybe it's going to be a little bit more clear why the next shrinkage is needed for this process, and maybe you can start to make start to think about when this process could occur. But we have a limitation of how fast we can image it. And we can image as fast as we finish the protocol, but the protocol is slow, right? And the protocol is slow because it's 40 repetition at 0.5 hertz, right? So that takes some time. So if you have something, if the plasticity occur in the middle of those repetitions, then you have, so we will have to image while we are doing the uncaging. Right, and for that case, of course, it would be great to have another set of galvos so that we can just, have another system that is imaging while we're seeing this, right? I don't know if that answered the question properly, but
yes,
I guess
yes. And another question: does initial spine neck length have any impact on LTP expression, either structurally or functionally?
No, so
do you see the question?
So, yeah. I mean, I didn't see it on the. So these are very good questions. So of course, if you have a spine that you cannot really see the neck, I think you're in a business that is impossible to really assess what will happen there, right? And I think there are some experiments that Sabrina and Diana in the lab did with spines that were borderline, and you can, I mean, we are in diffraction limited resolution scenario here, right? So, but the only thing I can tell you is that we always try to trigger plasticity in spines that have a visible or like a clear neck, right? Because otherwise we cannot really assess that parameter. Now, the other important question that is not directly that, but is the size of the spine head, which is also a very important aspect, because if you have a very big spine head, you might not trigger plasticity. So we deliberately selected spines that have heads that have been before observed to be successful triggers of plasticity or depression. So we confine our spine pool to spines that have, I forgot exactly the number, but spines that have been previously shown by other groups that are spines that successfully grow or shrink, right? And for the neck, spines that we can clearly see a neck, okay? I don't know if is that okay? You're welcome. Okay, sorry, I'm going too fast here. All right, so the same, we try an extra point for the LTD. And as I show you, this is a timing where we significantly trigger depression. But then if we separate them a little bit more, minus 23 means that there's 23 milliseconds between the initiation of the backpropagating action potential and the induction of the uncaging response. And so at that time, we were surprised that there was no LTD whatsoever, because previously it has been shown with connected pairs that the LTD window is a little bit longer in these conditions. So that might be related because we're completely bypassing the presynaptic, and any other, let's say, cannabinoid mechanism or whatever that might be making that window a little bit wider. But anyway, so in this particular case, we saw that there's one particular window that is very successful at triggering LTD when we activate a single spine with this post-pre-pairing protocol. Okay. So this is the rule that we uncover for a spine time independent plasticity at the level of a single spine. So we have a very successful timing that trigger LTP, pre-post timing, or this coordinated, if you with the same analogy you have, where the pre precedes the post. We have a significant neck shrinkage, then this timing is not good. Then at minus 13 or 15, it successfully triggered LTD, sorry, and a little bit longer times, nothing happened. And the morphology here is not compromised. So the induction of TLTP and TLTD in single spine follow this Hebbian STDP rule that is bidirectional and favor the presynaptic inputs that precede postsynaptic spikes and depresses presynaptic inputs that are uncorrelated with postsynaptic spikes at very precise and narrow temporal windows. Maybe that's not surprising, but I think because this is pretty much what other people have found, but it was pretty cool in my mind to crack it at the minimal functional unit of plasticity, which is a dendritic spine. Okay, so this is the same. So, and then we got, more ambitious is not the word, but we really wanted to expand this a little bit further, right? And as you probably know, STDP is a process that is dependent on the postsynaptic depolarization and ultimately on the level of calciums that you generate locally. So this is showing the same paper from Jesper and Mike Heuser. It's a beautiful work. And in this work, they just, as I show you, the pre-pose when you have a connected pairs of layer 5 pyramidal cells, their contacts are nearby in the pretty much proximal dendrites, then you trigger potentiation. But if you now have connected pairs of layer 2-3, which are a little bit high up, and layer 5, then you have the same pairing triggers depression. And the reason for that, when you look at the rise time of the synaptic connection between layer 2, 3, and 5, the rise time is a good proxy of where those inputs are being located. So now they uncover pretty much a rule in which the same pre-post rule, if you activate proximal inputs, trigger LTP, but if you start to go farther away, the same pre-post will trigger LTD, because there is this dendritic depolarization attenuation factor that will definitely affect your local voltage and change the same protocol, but now it's not going to be able to trigger potentiation, but it will trigger depression. In other words, telling that the local voltage deflections and local calcium are key for this process of triggering either LTP or LTD or nothing. And here is also more strengthened here by this experiment where they use extracellular stimulation, weak or strong. So if you have a weak response that triggers depression, you increase the strength of that connection and you trigger a potentiation. Anyway, so, but again, a direct demonstration of how synaptic cooperativity at the level of single spine is unknown. So what we decided to do is just say, all right, so we have here a particular timing that is very close to a timing that triggers potentiation, but when you activate a single spine, it doesn't do anything, right? It doesn't really care. So what if we add a nearby spine here? Can we just increase the voltage, increase the calcium, and as a consequence, enhance the timing window that is permissive to trigger LTP? So protocol, same here. We activate two spines before and after, and we use a timing 7 milliseconds. You remember was this timing that nothing happened. What we found is that if you activate these two spines, this is the EPSP that is being given when you activate these spines just themselves, without the pairing protocol. Then you do the pairing protocol, the 7 millisecond, the plus 7 millisecond, and then like about 9 minutes later, we saw that there's a significant shrinkage of the spines and a significant increase in the APSP. Just remember that at that time, if you do the same protocol in a single, nothing will happen. So just adding one nearby spines into the activation protocol, it changed drastically this timing to a timing that effectively can trigger LTP. Similar as was suggested by the paper of Jesper Sjöström, in the sense that the local voltage is very relevant for guiding the induction of LTP or LTD. So this is a curve, it's pretty straightforward. So the EPSP goes up and it remains up for the duration of the experiment that we tested. The necks shrink significantly and no changes in the spine head. So this is just for you to, for a rapid comparison. This is a 7 millisecond times, nothing happened, but the same timing, but with two spines, we can effectively trigger potentiation. All right, so now what else we can do with this? We can just now start to uncrack the code about what is the spatial distancing or the length constant that will break this cooperativity effect. So in order to do that, we just activate cluster, but then we start to slowly spread them out and see if now at that timing, we still keep triggering plasticity or not. And so here is one case where, remember, when we activate a 7 millisecond, it should have triggered a big potentiation. But if they're separated by more than 10 microns here, nothing happened. So we completely wiped out the induction of LTP by just putting those spines a little bit farther apart. When we look at the length constants, and we are looking at the max change either up or down of each of the experiments, we have this function here that gives us a lambda where, if normally potentiation is triggered when these spines are being clustered, it kind of disappears when they are more than 5.7 microns away. So we use that value of lambda to separate our data sets into cluster, which is less than 5 microns, and distributed more than 5 microns away. And what you can see is here is what I showed you before. This is the uncaging potential, this is the potentiation when you activate a single spine, nothing happened. You add one extra spine, you trigger the potentiation of this timing. You separate it more than 5 microns, you go back to nothing. And then the same happened with the neck. You have neck shrinkage only at the timing that you successfully trigger potentiation. Nothing happened with the head. So this is sort of what I mentioned to you. At the same timing, one spine, nothing happened. Two spines trigger potentiation. You separated more than 5 microns. You wiped out the induction of potentiation. Okay, so probably we'll try to go fast in this section because I think how I'm doing on time. I think I'm doing pretty.
It's 10 to noon, your time.
Okay, so.
And I have a question.
Okay, go for it.
So how do you account any receptor differences or synapse-related machinery?
Right. Absolutely. So that we cannot know the amount of receptor per spine. There's a good correlation, though, between the spine head size and the amount of AMPA receptors. So we don't know exactly how many receptors we have there. There might be variability, and in fact, we see variability in our data set, right? So so that's something it would be great to know in more details. But one way that we can minimize that is by just trying to look for, as I said, the spine head size to spines that are within sort of like a confined range of spine head volumes. Of course, I'm not going to these gigantic big spine heads because I'm not going to trigger plasticity there. Also, their impedance is going to be different. And so, yeah, but it's a question we don't really know. But you will see later that we look at the calcium signals directly at the spine heads and dendrites, and there are pretty clear patterns that explain when we trigger plasticity, you have X calcium signals, and when you don't trigger plasticity, you have this other type of calcium signal. Okay, so that will obviously represent the activation of AMPA and then NMDA receptors, allowing the calcium influx, some voltage-gated calcium channels, and maybe some other calcium sources. But that, in a way, is an indirect way for us to also look at what's happening locally in terms of voltages and potentially activation of glutamate receptors. Okay. All right, great. So this section, I'm going to try to go a little bit fast because it's more in the details of why we care about the neck, right? So why we have the neck shrinkage. And as you saw, the head in our hands doesn't change, but in the LTP, but the neck retracted. So initially, when I was doing my postdoc in Rafa Eustace lab, and then I worked with Tim Vogels with some models to try to explain if this change between that spine to that spine and this change in EPSP size could be purely explained by electrotonic solutions. And in fact, it can, but you will require incredibly big neck resistances in the order like tens of gigaohms, which is really, really crazy. So, but the other interesting output from that, from those modeling, is that the obviously this could be triggered very rapidly by only adding some few AMPA receptors into the spine. So we decided to test in our model why we have the potentiation. Is just a purely AMPA receptor mediated phenomena, or is the neck, is there some electrical aspect to it? We tried to tease apart these two questions. So what we did in order to ask the question about the involvement of AMPA receptors, or how these AMPA receptors are being added into the spine and maybe increasing the synaptic strength, we used a trick of putting this peptide inside the cell that it sort of competes with the GluR1 subunit of the AMPA receptors to interact with the PDC domain that basically anchors them to the postsynaptic density, which is the place that they receive the synaptic information. So if you do that, you basically avoid all this lateral diffusion and insertion of AMPA receptors, new AMPA receptors into the postsynaptic density. So we said, okay, let's do that. Let's see what happened with the plasticity. What happened is that a protocol that effectively triggered potentiation, we completely wiped out. If normally you have a potentiation, now we completely wiped out the induction of depression. But the surprising thing is that the neck shrinkage still happens. So we still see the neck shrinkage, but we block the plasticity, right? So we said, okay, so why is the neck shrinkage needed, right? So it's kind of puzzling, and I'm going to skip that because this is so we thought that maybe the neck shrinkage could be helping the AMPA receptors to laterally diffuse to the spine head. So the premise will be, and there's actually data that I'm not going to go into the details that basically show that, you know, the diffusion of receptors in spines with larger and thinner necks is harder than in spines with shorter necks. So the neck, if you redistribute the same amount of membrane, and if it shrink, it gets wider, and maybe that could just facilitate the Brownian diffusion of AMPA receptor to the postsynaptic. So that was the premise, but we didn't know. Or maybe it would be that just the neck doesn't really care, right? So we didn't know. So to try to break this neck shrinkage, we obviously focus in actin. Actin, as you know, is heavily concentrated in spines and primarily in spines. So we use this latrunculin A that basically binds to globular actin. So basically inhibit the polymerization of actin. So we thought this plasticity process, you're breaking this the sticks of actin, and then you want to repolymerize them to build a new scaffolding or whatever. So we just block the actin polymerization with latrunculin, and what we found is that we completely wiped out the neck shrinkage at a time in that we should have observed potentiation, and we not only block potentiation, but we also kind of diminish the response. So just messing up with the neck shrinkage, it completely blocked plasticity. So that led us to believe that basically suggests that LTP occur via mechanisms that involve the neck shrinkage-dependent facilitated diffusion of GluR1 subunit from the spine neck to the head and subsequent incorporation into the postsynaptic density. So we believe that this is a drawing I made. That you have this sort of actin filaments, then you trigger plasticity. There's calcium here. Those breadsticks start to break. Then you need to polymerize them to just build up a new scaffolding. But if this is impaired, the neck cannot shrink, and then the AMPA receptors cannot laterally diffuse and build up and be in the spine head. Of course, many experiments need to be done to completely corroborate that, but I think we very clearly show that the spine neck shrinkage, it is fundamental for the induction of plasticity, and the incorporation of AMPA receptor is fundamental for the induction of plasticity. So that's sort of the conclusion we get from those experiments. Right. So let's go back now to this rule and the cooperativity rule, right? So I just show you that if, or before the molecular mechanisms, I show you that if you activate an extra spine at this timing, you can just trigger potentiation here. What happens if we add an extra spine at a timing that you trigger LTD, right? And as you know, LTD induction in distal dendrites of layer 5 pyramidal neurons trigger LTD instead of LTP. LTD can be converted into LTP by increasing the local voltage. So the prediction will be if you increase the local voltage here, you might either completely wipe out the LTD or even trigger LTP by adding nearby spines. Protocol is now activating 2 spines before and after in this protocol, the pre-post protocol that successfully triggered LTD, but now adding an extra spine. And what we observe is that nothing happened to the spine head, but also we didn't trigger depression. If we only activate a single spine here, you would have seen a significant depression of the signal, of the signal given by this cell. But just adding one extra spine can completely wipe out the induction of depression of LTD. So this is very interesting because it's basically this is a cluster spine, so they have to be close to each other. We completely reduce the induction, and that tells us that by having coactive spines in a small chunk of a dendrite, it will bring your rule to an STDP rule that only encompasses LTP, because it gets rid of the induction of LTD. All right, so we had the opportunity to split the spines, or separate them, not split them, but separate them, activate them at different distances. And what we see is that if you activate here 2 spines that are very far, then you recover the induction of depression. And then when we look at the lambda, at the space constant, the length constant, sorry, we see that when they are clustered, you don't trigger depression, but then when you start to separate them out, you kind of recover the induction of depression. And the lambda here is different to the lambda that we observe with the cooperativity in LTP. So that means that there's something else. Maybe it's just a purely electrotonic attenuation that, you know, as you know, the lambda in layer 5 pyramidal cell is about 50 microns. So maybe the local voltage, when you separate it more than 30 microns, is not large enough to build up voltage and calcium to trigger the depression. And this is sort of when we, and then we use this lambda value as our splitting factor to spines that are clustered or distributed. So when we distribute the spines, we'll recover the induction of depression. When we look at the spines, here is the LTD. This is when you activate one spine at this time that successfully triggered depression. It triggers depression. You put cluster spines at less than 40 microns. You wiped out depression. Then you separate them out, and then you trigger depression again. And then, in fact, in this timing where we add more voltage by activating a single extra nearby spine, we even have a significant neck shrinkage. And we have some data, which I'm not going to show, but we look into the proximity of the spines. And with the more closer those 2 spines are, they not only wiped out the induction of LTD, but they go even into the induction of LTP. Okay? All right, so this is the rule that we uncover for the STDP at a single spine, a single cluster and distributed spine. So this is a classical bidirectional STDP rule. This line shows the boundary between the pre-post and post-pre and different timings. So we see that we have a very narrow timing when you activate a single spine that can trigger potentiation. Then if you have the postsynaptic leading the pre, you have depression. You add a nearby spine and you completely wiped out the induction of depression. And in addition, you increase the time window that is permissive for triggering LTP. So you go into this rule that only encompasses LTP. You split them more than 5 microns. You went back to this narrow LTP window, but it's not sufficient to recover the LTD. You split them more than 40 microns, and boom, you go back to a situation where you have a single spine. So in a condition where distributed spine and isolated spines, they follow the same rule. But then if they are clustered, It depends where they are located to enhance LTP or the same as before, but in both cases, it wipes out the induction of depression. And I show you about the biochemical aspect. All right, so the final slides, and I swear that it's going to be not more than 5 minutes. So we decided to look, and I think this is probably one of the most cool findings, because we decided to look at the local calcium signals while we're triggering STDP, either TLTP or TLTD, in all these different combinations. And we use the protocol I mentioned to you before about looking ROIs of the spines before and after the uncaging. So what we found is really surprising is that this is, just bear with me, this is repetition 1. This is, we are imaging the dendrite and the spines that we're activating throughout the induction protocol. So there are 40 repetitions because we have 40 pairings, right? So this is the before image at repetition 1 is the baseline. And then we look at the image, after the uncaging, and then at the basal before the second repetition, and we go back and forth. Here is an ROI, so we cannot really look precisely at the peak of the calcium right after the uncaging. Sometimes we pick it, but we are trying to focus on the accumulation of calcium during this protocol. And what you can see here, calcium goes up in the spine and in the dendrite, but then in the next repetition, after these 2 seconds, it goes back to baseline. It goes up, to 0 to baseline, up to 0 to baseline. So in other words, at this particular timing that is not affected to trigger plasticity, the calcium goes up, but it goes back to baseline before the next repetition. When you activate an extra spine, what happens is that the calcium goes up in the 2 spines in the dendrite, but in the next repetition, the calcium is still up. Then it goes up, and so basically you're going into this like accumulation of calcium, which is absolutely incredible, because here you see in the spine, this is before the pairing protocol, so this is just focalizing on the baseline, and you see that in one spine, pretty much it always goes back to baseline, but you add only one extra spine and it builds up. Okay, this is sort of the same, but taking the spine after, so we have also this significant accumulation. And this is actually causing the plasticity. In fact, when we block, and I'll show you, I think it's here, when we block NMDA receptors, we completely wiped out the accumulation of calcium, and we completely wiped out the induction of plasticity. So it's an NMDA-dependent buildup of calcium in spines. Same at the spine, the single spine that we trigger at plus 13, the same process happened. There's accumulation. In this case, it's just one spine, but the timing allowed the spine to accumulate calcium and trigger the plasticity. For the LTD, and I forgot to mention, maybe all of you know, but there is pretty well established that the induction of LTD and LTP depends on calcium. And there's this calcium hypothesis that basically, lower calcium levels in the spine will trigger LTD, but then if you start to go up, you will trigger LTP. So it's the same molecule that can trigger both processes. So when we look in the post-pre timing, we saw, I think it's interesting to see that it's kind of started, here. So basically, it kind of remained in this level. The calcium goes up, but it kind of remained on this not so high level, but still some calcium there. And so in other words, what we have is that there is also some calcium accumulation, but it's not much, but it remains in a very lower level or state. So let me just wrap it up with this. So this is what I show you with the rule about the induction of plasticity. And the calcium control hypothesis is something that we are proposing. I mean, this is a hypothesis that has been proposed before by many, but I think what we're providing, which is, I think, rather new, is that we need to accumulate. It's not much, it's about how much you accumulate during your induction protocol. So in other words, if you have a situation where you want to trigger LTP, your spine, for example, at a permissive timing of the trigger LTP, you start to accumulate calcium until you pass the threshold for triggering LTP. LTD probably remains in this state, which is permissive, which has a much narrower window of start and stop to trigger LTD, and other conditions where you don't get there by, let's say, when you don't have enough of sufficient calcium. So the conclusions of our work is that, we really show that there's a structural or it really matters how your synaptic inputs are being impinged onto your dendrite to either trigger classical bidirectional Hebbian STDP rule if they're distributed or single, isolated, or there's conditions where you have cluster inputs that you're going to go into a rule that will only encompass LTP. And that probably will make a ton of sense because the rarity of those events might be rare to have these cluster inputs activated at the same time, and maybe there's a command there to either do nothing or only trigger potentiation and not erase that information. We look, as you said, at many of the molecular machinery going on there, the role of next shrinkage. And I think what we like to propose is this concept of microclusters that describe this structural and functional modality of synaptic connectivity. And as you know, microclusters have been observed before. And there's data here from Schmidt et al. And that they have seen that there are a series of axons in one axon that can contact 2 or 3 spines. There's a cluster, there's a presence of cluster connectivity right in this innervation of the same axon, and it's a space of less than 10 microns in the basal dendrites of layer 2-3 pyramidal neurons from middle to renal cortex. So you can have the same axon impinging 2 or 3 spines, which obviously will activate them simultaneously, and maybe will build up a memory there. And there's these other papers with by Hoffer and by David Fitzpatrick, which they showed this very beautiful. They show these orientation bars in an awake behaving animal, and they see that the orientation selectivity is basically confined to these microclusters of spines. So in other words, these microclusters, share receptive fields properties. For example, these 3 spines here, which are only these 3 spines, respond to only this orientation bar, and so on and so forth. And when you look at this and the tuning correlation versus the dendritic distance, the lambda of that is about 5.3 microns, pretty similar to what we saw with the condition that maximize the induction of potentiation. Same here, this is the spine distance. They also calculated lambda, and it's also about 5 microns. So with this, I just want to leave you by saying that I think there's a very important reason why these microclusters that have been found before but have not been really understood in the context of their role in building engrams, and they can be gated by the convergence of different axons, which should increase the computational power, or by by same axons or by different axons. And I think the lab of David Fitzpatrick have shown very beautiful data showing that sometimes different axons can trigger cluster spines. And as you will know, in vivo images of dendrites during active behavior, active task, you see a bunch of these clusters popping out over and over and over. So the reason of this might be that if you have those microclusters, then you will try to build a memory there. Finally, probably the most important slide, and the team, these are the 3 fantastic researchers, very, very strong, smart, and talented people, Sabrina Tasserat and Diana Mitchell, the first co-author of the paper, and Soledad Miranda, that did all the molecular biology of the actin machinery in the spine. And with this team, and only with resources from the Canadian government, and with the fantastic help of Mohamed Nasir, that he really helped us to push our microscope to things that we were not even sure we could do. Paul Reesen and of course Mike Barrett, that really helped me to set up something which allowed me to do all this. So thank you very much. We'll take some questions. Maybe I went a little bit too far, but in time. But anyway, thanks so much.
Thank you so much, Roberto. You captivated our audience. We kind of over time, so I will read 3 questions that are left. So Neil would like you to speculate about, I think this one, yeah. Why do you see only a shrinkage of the spine neck, but no change in spine head?
Yeah, so we really dig into that very thoroughly because as you know, many great work by Carencito and by Ryohei Yasuda and many others have shown a spine head enlargement. And I think in many of those cases, what they do is that they remove magnesium or lower the magnesium concentration so that you have NMDA receptors active, right? So that you can just speed up that process to trigger this consolidation or this buildup of a big spine, the plasticity. So on the spine head enlargement, we see it when we remove magnesium. So we did the experiments with the same protocol that we used, but we removed the magnesium and then we saw precisely the head enlargement. So it's not that it doesn't happen, but in our case, we wanted to keep the magnesium untouched and we wanted to trigger something that will be as physiological as we can. Of course, these are all mimicking certain things. And but again, I reiterate, the head enlargement happens, and there's many papers that have seen or shown that spine head enlargement occur in the awake behaving animal, right? So that happened. I guess what we are proposing here is that we're encountering or we are describing a new earlier stages of LTP. So in other words, you have a spine that is ready to be on the plasticity. It gets this potentiation, it gets into plasticity, and maybe later on you can go into a protein synthesis dependent or something that will consolidate that spine even further and will increase the spine head size. And by that, you will basically change the impedance of the spine. So you will transform it in a very reliable spine. So the head enlargement has a lot of value to it because it can just basically transform it in a very low impedance structure that is primarily a glutamatergic synapse and doesn't engage many voltage gated channels and so on and so forth. I hope that answered your question, but short answer, yes, we can get head enlargement by removing magnesium.
Okay, thank you, Roberto. So I have one amazing talk. Lillian is saying, amazing talk. I was curious about the location of the uncaging. Does the timing of LTP, LTD depend on the location of the synapses?
Right. So this is a great question. So and I don't know, maybe I passed the slides a little bit too far, but there's a somatodendritic dependency on the STDP. So in other words, if you're very close to the soma, when you generate a backrotating action potential, it's going to take only little to go like 80 or 50 microns to a spine, right? But if you have a spine that is very far, the backrotating action potential, by the time it gets to 80 microns away, the voltage is not going to be the same. So that's why a protocol, a pre-post protocol that normally trigger plasticity in proximal dendrites, it will most definitely trigger depression on the very distal dendrite. We have not done that yet. And one of the main complications for that is that in addition to all the complicated experiment that we will have to have a dendritic patch reporting the voltage locally because a single spine you will not see it at the soma. You see what I mean? So in other words, yes, I think the main message here is that it's all about local voltage, local calcium. You can get that with backrotating action potential, you can get it with dendritic spike, you can get it with whatever that is activated there. And basically you are accumulating that calcium, and at some point you reach the trigger LTD, it's going up, you reach the trigger LTP. So that could be, as I said, triggered by you can think of many combinations of synaptic activity and dendritic activities that can get you there. And this is sort of what we're trying to do now, to try to tease apart the most essential logic of this flipping between one condition and the other, and what you need to have in order to get one or the other. Yeah.
Thank you. And then, so all this work is done in vitro. There is a question about how this rule applies to in vivo activity, where the animal, is stimulated outside, more spontaneous.
Right. So I mean, yes. So this is all in acute brain slices. We would love to do it in vivo. There's some protocols out there. I think Miran Kasur has a very interesting paper where he's triggering some spike timing dependent plasticity kind of thing with the visual cortex, and he's using the spine head enlargement as a proxy of induction of plasticity. So there are ways that you can do that. They're very complicated, and we're thinking on those terms, right, to try to see that. But one first question that we might want to ask is the presence or absence of these microclusters, right? Because if by definition, if you believe everything I said, you will say, okay, come on. I mean, if you have two spines, that's really crazy, right? I mean, one extra spine nearby can change the logic. Right, but there are conditions, there's the release probability things that you have to play around. But what we really want to do, and David Fitzpatrick has been doing this for many years now, but is to look at the dendrites while the animal is doing some behavioral relevant task and see what is the propensity of having that representation of inputs related to a meaningful task, for example, versus not. And so you can start to tease apart the question of how your dendrites are being activated, just without trying to trigger anything. What is the pattern of activation of those dendrites? And maybe that can start telling us also more about the what we need to look in the dish about those particular rules. But yeah, so we're trying also to develop in vivo uncaging strategies. So we eventually will be able to try to emulate the same type of things in vivo and see whether this rule holds true as well. But it's a good question. We don't know yet.
Thank you. And we have another, Shane is asking about some mechanistic approach. So he says the release probability of a synapse is typically less than 100%. Have you performed any experiments with a stochastic where the backpropagating action potential coincides with spine activation in 30-50% of events?
That is a great question, and we have really thought deeply about that. So, and I think maybe the way to answer that question in the way we're thinking about it is to look at the calcium, right? So as I show you in the calcium, we see that the calcium locally, is building up. So we're doing Fourier repetition. We might not need Fourier repetitions, right? And we might need not maybe 0.5 hertz. We might need a different frequency. So the things that you can change, I think the bottom line to me is when you get to the calcium threshold that is needed to trigger A, B, or C, right? And so now we have a very standard protocol that allow us to see the breaking of this rule by just adding a spine, right? If you make it a 1 second separated time points, you will not have the accumulation, and we would have not seen that breaking of the rule most likely. So these all tell us that it's not. There are going to be many roads that will get you to Rome. But the point that is screaming loud is the calcium level locally of the accumulation, how you get there. And that's what we're trying to go at. You're right. It's like the most, it's a stable situation we can get. But luckily we did it like that, because otherwise, as I said, we would have not seen the breaking of the rule by adding an extra spine, I would suspect. So I'm very happy we did it like that. Yeah.
I think this is gonna be it, right? Thank you so much, Roberto. Inspiring talk, many questions. Thank you so much. Thank you all who stayed with us for so long. But if you have any additional questions, there is email available. Please send us emails, and also if you have any ideas for next webinars, the topics that you're the most interested in, I will be happy to invite our customers. We have a broad range of customers studying everything brain related. So I would like your input on this one too. All right, I'm finishing up. Thank you so much again, and I will see you next time.
Thank you very much, Eva. Thank you very much to the Bruker crowd for helping me make this project a reality. So I appreciate it.
Thank you for your time, Roberto. You're special. Thanks.